Hi, I'm Jason Zhang. I'm from University of Washington. I'm a postdoc there. I would say that in the beginning, when I was, growing up, knowing that I was LGBT, I always was interested in learning about HIV because of the associations with that community and HIV. So that was one of kind of the first introductions of me interested in biology. And I knew I wanted to do biomedicine, but I also knew I hated blood and I hated needles, so I could not be a medical doctor. And coming from my family, we're a family of engineers, I was thinking about bioengineering. So that was what led me to that path. But what really got me interested in pursuing an academic career in science is when I was doing grad school and I did some experiments and saw for the first time that sometimes you can see the first evidence of some new thought in terms of this field, and just getting that like kind of new hot off the press data really made me excited about trying to conceptualize what does this mean and how does this could this change the way that we think. And I think that just spark of discovery really made me interested in really pursuing academic science. So I'm generally interested in signal transduction and understanding this concept of a phenomena of molecular pleiotropy, which is where a single molecule inside our cells can have multiple different functions. And so understanding how does a cell know whether to do function X or function Y. And it's really important for signaling molecules because for signal transduction, you have to dynamically alter depending on your environment. So how does a cell can dynamically alter these and their functions in order to be appropriate for the external stimuli? So my general hypothesis is that spatial temporal organization of these signaling molecules within a cell helps dynamically regulate them and be able to respond to these external stimuli. And in order to probe into this, what I do is use protein engineering and protein design tools, along with computational tools, in order to build new and expand the molecular toolkit for making sensors and perturbators of this organization system, signaling organization systems inside cells. And this also has broad implications in terms of diseases because a lot of diseases, such as cancer, have this signaling organization disrupted. And so, therefore, trying to understand both the basic principles and also the translational kind of consequences of this intricate signal organization inside cells. I think the cool thing is that the more like these pathways are so ancient, I don't want to say ancient, but so well studied. And we studied for them for decades. And it's always interesting that even though we first think of that these pathways being going from point A to point B, there is the more you study it, the more complex it becomes. And it becomes can, either be really a lot to take in, but also could be really exciting to really understand, these classical pathways. There's still a lot to learn from them. My educational path is quite linear for the most part, but there was some twists and turns in terms of um, trying to explore what I was really interested in biomedical research. So I initially thought I would do biomaterial research where I would be working on, for instance, tissue engineering, but then explored in my rotations during my PhD quantitative biology, and I thought that was really interesting and really satisfying as well, so to understand the mechanisms. And there's definitely times where I didn't even know if I would even could get a PhD, and times where I doubt myself, but definitely through the wonderful mentors in my PhD at UCSD and also at University of Washington, and for my postdoc, um, they definitely helped me try and realize that science is of course not linear, but there's always a journey, and the part of that journey is part of the fun of it. Right now, I'm exploring being an academic faculty. So I have applied and are going to apply and within these coming cycles. But what I'm really interested in is kind of like my pitch, my overall kind of like one title pitch is to bring protein design into cell biology and to really both focus on the questions I'm interested in spatial temporal signaling, but also use it making more accessible these protein design tools and protein engineering tools to the scientific community and helping people also expand the molecular toolkit for their question of interest as well. So that's my general scientific interest in my future lab. I love the fact about the sparking discovery, and even though this might be bold to say, but always thinking about rewriting the textbook, and I think that's just a cool phrase about um, just thinking about how we think science is a very kind of like this is the way things are going to be, this is the truth, but then a lot of times there are a lot of good data and good facts as well, but there's also more and more complexity in science, and I think that's just fascinating to understand more and more about these and trying to expand our knowledge. And in terms of like advice for other people, even though I feel ill-equipped to really give advice as well, is that I always find it most interesting when I go to like seminars and conferences with like for people who are doing kind of still within the realm of biology, but like kind of slightly different fields. And I always find it fascinating how they answer a scientific question, and it definitely brings inspiration to for me in my research. Like, oh, maybe I should think about the question in this way, or maybe I should utilize this tool that the immunology field or the neuroscience field uses a lot. So I guess that all comes to a wider kind of like aspect of that. There is always exploration around science as well, and that what I thought I would be doing in terms of when I first started grad school is totally different than what I ended up doing and continuing doing, and what I found fascinating. So I think that there's always a lot to explore, and that could be daunting as well, but that it can also be exciting to just see what's out there, and then hear different perspectives, and it'd be great to have different perspectives on the same problem, and have a kind of collective hive and trying to understand these really fundamental questions in biology.